In this video, we're going to talk about the phylum Porifera, and basically, we're talking about the biology of a representative Porifera, and that is Leucosolena. Now, what are Porifers? So basically, Poriferas are derived from two Latin words, that is porous and fera, which means pore bearers. So Poriferans are pore bearing species, and they are basically classified as plants not until 1857. Not until 1857. That is where they were classified as animals to be the what? To be the remnants of some groups of metazoans. So they are the most primitive animals that includes about 5,000 species, and they are the first multicellular animals that have a pore, and the pore is called ostia. I mark it, called ostia. Now, basically, let me add this. Basically, in the multicellular animals, sorry, I have a very good handwriting. Those that knows me knows that I have a very good handwriting. So multicellular animals can be classified into two. The first one is the porifers, that's the sponges, and the second one is the eumetazoans. The eumetazoans. You understand? So these are two classification of multicellular organisms. So basically, we'll be talking about the poriferan first. You understand? I hope you can see my handwriting. We'll be talking about the poriferan first. Now, let's talk about um the advancements of porifers compared to the phylum protozoan, because remember we talked about the phylum protozoan before. Now, what are those things that porifera used to be advanced more than what? More than the phylum protozoan. Now, the first thing is their size. The protozoan have a very small size, therefore the cytoplasm can have the limit that um the cytoplasm have the limit of the nucleus it can control. They have a very small size. You understand? So once they attain a very large size, they do what? They break into two during using binary fission. So that is their size. While the poriferans, they are what? They have a very large size compared to what? Compared to compared to protozoan. And I can also say, because of their small size, protozoans are microscopic. Hmm? They can only be seen with the aid of what? With the aid of microscope. While porifers are macroscopic. You understand? They are all macroscopic. Now, what about their sexual reproduction? The sexual reproduction of this group of animals, this binary fission, and their sexual reproduction of this one is what? Is by budding. So it is quite different. I'll tell you what a budding is. Now, the complexity of the system. The complexity of this one is they are less complex, and the complexity of this one, they are what? They are more complex. While the level of organization, this one is in protoplasmic level of organization, while this one is in cellular level of organization. Talking about their surface area to volume. Talking about their surface area to volume, this one has a large surface area to volume. Hence, they can transport material with the aid of simple diffusion because of large surface to area volume. But this one, they have a very small size area to volume ratio when compared to what? When compared to protozoan. Are you getting me now? Now their cells, these ones, they are all unicellular. All they are what? They are all unicellular. While the phylum Poriferans are what? They are multicellular. Are you getting me now? So in other words, in a simple term, we can call Poriferans, we can call them sponges. Sponges. In a simple term, we can call them sponges. And remember, I said we derived their word from two Greek words, which is porous and feran, that is porifera. You understand? So moving from there to the general characteristics of, I've talked about this already. I said they can be classified into two. That is the poriferans and the hue metazoans. Hue derived from what? From hue means through, through metazoan. That is the meaning of hue metazoan. Through metazoan. You understand? So that is where we derive the word hue. For poriferans, They are what? They are still in cellular level organization, even though they are what? They are multi-cells. Now talking about the characteristics of what? Characteristic of poriferans. The cells of poriferan are loosely organized rather. They are what? They are loosely organized. Hence, I can actually say in my own word, I can say their cells are versatile. And when they see something is versatile, it means they perform a particular function, but still the function of this, like the function that they perform, can change with time. Hence, they can attain what we know as division of labor. I think the ink is too thick, so let me just reduce it. So they can undergo what we know as what? Division of labor. Are you getting that? So now the second thing is most of them are found in marine water. I have to note that most of them are found in marine water, while few are found in freshwater. They exhibit two types of symmetry. Either they are radially symmetry or asymmetrical. That is non-symmetry. Non-symmetry. Is either they have a radial symmetry or they have no symmetry. You understand? Now, the next thing is their body is usually cylindrical. That is their body shape. And they secrete what we know as scleroblasts, which secrete spicules, while spongin fiber are secreted by what? By spongioblasts. I'm telling you about all these cells. Now, they have no organs in their body. So, like I said, they are in what? They are in cellular level of organization. Are you getting that? So, they are in what? They are in cellular level of organization. And their body comprises of numerous pores called ostia and osculum. The pores are known as what? Ostia and what? Osculum. And they have a central cavity. The central cavity called the spongocoel or the atrium. But mostly they use the spongocoel as the central cavity of what? Of these animals. And when we talk about Leucosolena, we still go to another central cavity known as paragaster. Now, moving from there to other characteristics. Now, we said they reproduce sexually by budding and fragmentation, like I mentioned the other time, and they reproduce sexually by fusion of gametes because they can reproduce sexually as well, apart from asexually. I don't think I actually mentioned that here, but they can reproduce sexually as well. You have to note that they can reproduce sexually, though we'll talk about that in the biology of Leucosolena. Now, their nutrition is holozoic, which means holozoic means heterotrophic. Heterotrophic. They have heterotrophic mode of nutrition. And their neurosensory cells are devoid of specific nervous system. So they do not have a well-developed nervous system. And they have a power to regenerate the lost parts. They have the what? The power to regenerate the lost parts. And I will tell you about the cell that is responsible for what? For regeneration, and it is called amoebocytes. That is the name of that cell. Now, the development is indirect and the cleavage is holoblastic. They exchange respiration gas by simple diffusion. By simple diffusion. That is how they respirate. That's how they respirate and that's how they excrete. Now let us talk about classification of porifera. They can be classified into three. The first one is calcare. The first one is calcare. The second one is hexatellinidae. And the third one is dermospongia. Now, what is the difference between these three? What's the difference between these three? Now, for calcare, they are found in marine, shallow, and coastal water, and their skeleton is composed of what? Of calcareous spicules made up of calcium carbonate. The body is cylindrical and exhibits radial symmetry. The body is asconoid, synconoid, or leuconoid. Please note that it is either asconoid, synconoid, or leuconoid. Are you getting that? Now, of this calcare, demospongia, and what and this, they still have what we know as the spicules. They are spicules. The spicules of this one can either be mono or tetrahexon. I did not mention that here. Tetrahexon. Tetrahexon. You understand? Either monohexon or tetrahexon. While in the other ones, while in the hexatellinate, from the word hexa, it means they have what? They have hexahexon. Hexahexon. Are you getting that? Or they can have trial, axon. So either six or what? Or three. You understand? These are what they actually possess. While for Demosponge is the same as what? Is the same as the calcary. You understand? And talking about this Demosponge, about 95% of sponges, 95% of sponges belongs to Demosponge. You understand? 95% of sponges belong to what? Belongs to the Demosponge. And their body structure is always their body structure is always leuconide form. It's always leuconide form. We talk about that leuconide, asconoid, and synconoid form. So that is the what? That is the characteristic of what? The characteristics of the classification of sponges. I hope you get that. Now move from there to the next slide that talks about the types of canal system. We have three type of sponges based on canal system. Either it is asconoid type, the synconoid type, or the what? The leuconoid type. I get me now. So these are the what? These are the type of what? Of sponges that we have. So talking about the asconoid type, talking about the asconoid type, they have flagellated spongo coil. They have what? Flagellated spongo coil. But these ones they have flagellated canals. Are you getting me now? This one have flagellated canals, and the last one they have flagellated chamber. Now, one of the differences between this one is that they have spongo coil. You understand? This one also have spongo coil, but they have something we know as porospile, uh, prosopile, prosopiles. You understand? And this one, they also have prosopiles, but their spongo coil is completely obliterated. They do not have spongo coil. And they are the most complex. You understand? They are the what? They are the most complex sponges. You understand? An example is Spongilla species. So let me just draw this. Uh, let me just see if I can actually draw this. Let me clean this. Let us clean this, and we should draw this. So let's have something like this. So this is it like this. So let me write. I write that is the highest. Sorry. Let me clean this. Sorry. We have from the lowest. That is the lowest complexity to the highest complexity. So the most complex is the leuconoid, and the least complex is what? Is the asconoid. You understand? So we have three types of canal system: the asconoid canal system, the leuconoids, and the what? And the synconoid. You understand? So these are the what? These are the classification of sponges. Are you getting me? Now, moving from there to biology of Leucosolena. Now, so the what? Biology of Leucosolena. Now, the Leucosolena are referred to as primitive simple sponges that are found attached to objects, and the object that they are attached to, this object is referred to as substratum. Substratum. Hmm? Substratum. So that is the object that they are attached to. Now, substratum can either be seaweed, shells that is usually in a shallow water such as seas and oceans, and they are usually found as a tubular compartment. Each individual creates a vase shape with a central cavity called the spongocyst, the spongocoel, or the paragaster. The spongocyst, the spongocoel, and or the paragaster. So let me just illustrate something real quick. Let me reduce this. Just have something like this. Yeah, let us illustrate this real quick. Let us say this is a sponge. You understand? Let us say this is a sponge with all this thing like this. Let us say, for example, this is a sponge. Sorry, let me just clean this. Doesn't have this. Let me draw a tube like sponge. You understand? So let me say this is a sponge. Then it's attached to something at the bottom of the sea. You understand? Now, in this, we have this one to be substratum. We have this one to be substratum. That is what it is attaching to. Now, they now have an opening that serves as mouth. They have an what? An opening that serves as mouth. And that opening is termed osculum. You understand? The opening is termed what? Osculum. Remember, they have pores all over their body. Let me use thing to represent that. Alright. They have pores all over their body like this. Can you see? Something like this. Pores all over their body. And this pore is termed what? The pore is termed ostia. Ostia. And ostia is produced by porocytes. That is the cell that produce ostia. Porocytes. You understand? Now, inside the ostia, we now have an opening. The opening is called osculum, and the osculum serves as mouth. You understand? The osculum serves as mouth for the porifers. Now, moving from there to the layers of sponges. We have different layers for sponges. We have the three layers. Let me just, we have layers for sponges. We have layers. Let me say layers. We have the outer layer, outer. We have the middle, and we have the inner. Now, the outer layer is called pinacocytes. The outer layer is called the pinacocytes. The middle layer is called the mesohyl, and the inner layer is called the choanocytes. I think I wrote it somewhere around there. Okay, this is it. Look at it. The outer layer is called the pinacocytes or the pinacoderm. The middle layer is called the mesohyl layer, and the inner layer is called the choanocytes or the choanoderm. You understand? Now, the skeletal material, they possess a skeletal material. The skeletal material of these animals are needle-like or triradiate spicules or calcium carbonate. While in other sponges, we can have something like silica or organic fibrils, you understand, called spongin. Now, talking about their nutrition, they are suspension feeders. Sponges are what? Sponges are suspension feeders, and they feed on food that is suspended on flowing water and contains fine material. So, what have we said so far in this video? We talked about the what poriferas is, and we talk about their characteristics, and also we talked about the talk about the relationship or the advancement of Porifera compared to protozoan, talk about the general characteristics of protozoan, and we've talked about the types of classification of protozoan as well as the types of canal system, and we have talked about the structure of protozoan. And now we are talking about the biology of leuconoids, in which we will continue in the next video. Thank you for watching.